Very good afternoon to all of you. Yesterday, we have seen a part that was associated with antigens and some of the properties we have already discussed in terms of antigens. Now, let us continue with the second part of this particular lecture in terms of antigens. That what are the specificity of the antigen we have already seen, but now what depends upon for that antigen to start an immunological response. So, let us understand the antigens in a very perfect manner now. Correct. So, when I understanding this antigen, one has to understand that the antigens are very much specific in terms of their dose as well as in terms of the reactants that they work on. So, the dose of administration of an immunogen can increase the immunogenicity. There is a dose of antigen above or below which the immune response will not be optimal. So, in order to induce an immunological response, the dose of an antigen, the amount of antigen that comes inside the human body is said to be very much important. For an inefficient dose will not stimulate an immune response either because it fails to activate enough lymphocytes or because it induces a non-responsive state. So, very less amount of antigen is there, the cells of the immune body may not be able to recognize this in a proper way, and therefore the non-responsive state of an immunological response will be developed. And an excess amount of high dose also can fail to induce an immunological response because it causes lymphocytes to enter a non-response state. So certain lymphocytes will not respond in that way, because if the level of or the amount of antigen is very high, then certainly the non-responsive state would start. Now, route, how it comes inside: intravenous, into a vein; intradermal, into the skin; subcutaneous, beneath the skin; intramuscular, into a muscles; intraperitoneal, into the peritoneal cavity. So the administration route determines which immune organs or cell population will be involved in the immunological response. Antigen administered intravenously is carried first to the spleen, whereas the antigen administered subcutaneously moves first to the local lymph nodes. So ultimately, based on the insertion of that antigens to these various routes, the antigens may be classified or may be given to specific different organs in order to process that particular antigen and start an immunological response. Adjuvants, substances that are mixed with an antigens or injected with it, serves to enhance the immunogenicity of an antigens. Means ultimately these adjuvants will help to develop the antigenicity of a particular molecule. They are often used to boost the immune response, which an antigen has lower immunogenicity, or when only small amount of antigens are entered inside the immunological system. So the level of immunogenicity or level of antigen is too small, then these adjuvants sorry, get attached to it, and ultimately it helps to increase the immunological responses. Aluminum potassium sulfate acts to increase the antigens persistence. When an antigen is mixed with alum, the salt precipitates the antigen. Injection of this alum precipitates results in a slower release of antigen from the injection site, so that effective time of exposure to the antigen increases from few days without adjuvants to several weeks with the adjuvants. So ultimately, when the injection of aluminum potassium sulfate helps in, or alum is being mixed along with the adjuvants, then along with the antigens, then the immunological response would start. Fluence antigens. So what are these fluence antigens? There are two types of fluent antigens. Water and oil adjuvants also functions to prolong antigen persistence. Fluent antigens, incomplete antigens and complete fluent antigens. Fluent antigens contains antigens in aqueous solutions, mineral oil, or emulsifying agents such as manamide monoleate, which disperse the oil into small droplets during the antigen, and the antigen is then released very slowly from the site of injections. Fluents, complete antigens, heated killed microbacteria in the water in oil emulsion. In addition to a muramyl dipeptide, components of the microbacterial cell wall activates macrophages and phagocytic cells. So these are the different types of fluids antigens that are associated, which are artificial antigens or which can be designed in a laboratory and can then inject inside the patient in order to start an immunological response. As far as alum is being concerned, alum and both fluid antigens stimulates the local chronic inflammatory response, which are increasing phagocytic cells. So inflammatory response are also being activated with the help of these adjuvants. Okay. Now, epitopes. What are epitopes? In order to recognize the particular antigen, there are specificity. An antigen is a part of an antigen that is recognized by the immune system, specifically by antibodies, B cells, and T cells. These are known as epitopes or antigenic determinants. These are called as epitopes or antigenic determinants. They have been called as. B cell recognizes different epitopes on the same antigenic molecules. In the case of protein antigens, an epitope may involve elements of primary, secondary, tertiary, and even quaternary structure of the particular protein. In the case of polysaccharide antigens, exists side chain branching via glycosidic bonds affects the overall three-dimensional conformation of an individual epitope. So this designing of a particular epitope and the immunological response on top of it is said to be one of the important points that is associated to start a immunological response. T cell, B cell exhibit fundamental differences in antigen recognition. See here how they recognizes their the specificity in terms of a particular antigen. You can see this B cell. This is the antigen of a bacterium. Here, the B cell receptors is there, and on the B cell itself, once it is been recognized by the B cells, then the B cells will activate the immunological response, and the plasma cells and memory cells are being recognized. So, recognition of a particular antigen is said to be very different for T cells as well as for B cells. Mitogens. So, what are mitogens? Mitogens are agents capable of inducing a division in high percentage of T and B cells. So, mitogens are a little bit different. They are helpful for multiplying the T and B cells in a very large amount. Mitogens can activate many clones of T or B cells irrespective of their antigen specificity. Hence, they are also known as polyclonal activators. So, these are only activators of the immunological systems. They do not mount or they do not have a very pathogenic effect, but they are helpful for activating the immunological responses. For example, mitogen, what are the examples? Several common and mitogens are sugar binding molecules called as lectins. So, there are glycoproteins in there. Lectins molecules, membranes, glycoproteins often lead to agglutination reactions. So, maximum agglutination reactions are being generated with the help of this mitogens. Preferably activated B cells, some activate T cells. Preferably B cells are being activated with the help of mitogens. The most common lectins mitogenic activities are concavalin A, phytohemagglutinin PHA, or coquid mitogens. So these are specific mitogens which are helpful activating the T and B cells. Specifically, the B cells get activated because of the mitogenic activity. The lipopolysaccharide component of the gram-negative cell wall also functions as B cell mitogen. So there are certain bacteria also which have this ability to activate the B cells. The mitogenic activity of lipopolysaccharide is due to its lipid moiety, which is thought to interact with the plasma membrane, resulting in a cellular activation signal. So the LPS of the lipopolysaccharide plays an important role inside the bacterial cell wall, which helps in activating the B cell mitogens. Superantigens. What are superantigens? They are the most potent T cell mitogens known. Superantigens binds to residual V region domain of the T cell receptors and the class 2 MSC molecules outside the antigen binding clip. So ultimately, these superantigens are very specific towards their binding, and once they bind, they are helpful for activating. The common superantigens include Staphylococcus enterotoxins and toxic shock syndrome, which is produced by gram-positive bacterium Staphylococcus aureus. So these superantigens are helpful for activating a higher level of immunological response. Basically, it will again be helpful for the activations of T cells instead of B cells in order to deactivate the specific proteins that comes inside the immunological systems. So that's all for today. We have seen the types of antigens and the mitogens as well as superantigens.